All right, hello everyone. So this series of videos is going to review and supplement content from Chapter 2 of your textbook, which provides an introduction to cognitive neuroscience. Okay, so in this particular video, I am going to talk about a concept referred to as specialization of function. And actually, the next three of my lecture videos are going to focus on the idea of specialization of function. But in this particular video, I'm going to talk about specialization of individual neurons. Okay, so this video is going to focus on individual neurons, and then my next two videos are going to focus on the specialization of different structures of the brain. And then my last video is going to talk about something called distributed representation. Okay, so these are all concepts that you guys, of course, read about in your text, but I'm basically going to review those particular concepts for you as best I can. Okay, so let's go ahead and get started with specialization of individual neurons. Okay, so just to start kind of at the beginning, what is cognitive neuroscience? Cognitive neuroscience is formally defined as the study of the physiological basis of cognition, right? So what does that mean in concrete terms? We're basically just looking at what happens in the brain, right? So we're looking at decreases and increases in electrical activity of individual neurons or large assemblies of neurons in the brain. And we can do that using a wide variety of neuroimaging tools, which you guys have already reviewed in the previous video. So we can stick someone in an fMRI scanner and look for oxygenated blood flow as they're completing some sort of activity. We can hook them up to an EEG, which looks at neural firing of individual assemblies of neurons. We can do PET scans, which also looks at patterns of brain activation while people are completing various activities. There's all sorts of tools that are available to cognitive neuroscientists, but basically the overarching goal of cognitive neuroscience is going to be to investigate what is happening in the brain. What are the metabolic and electrical changes in the brain that occur when we're engaging in various aspects of cognition, whether that's whether we are attempting to remember something, whether we are engaging in problem solving, whether our attention is being sustained on a particular stimulus, whatever aspect of cognition it is, we want to look at the physiological changes or the changes in the nervous system that accompany that. Okay, so what does specialization of function mean? Well, specialization of function is essentially an attempt to answer this question, which is, are individual neurons, groups of neurons, or various structures in the brain specialized or localized to carry out various cognitive processes? Right? So can we point to specific neurons or specific brain structures as the neural substrates, if you will, of a particular cognitive process, right? Can we pinpoint individual structures in the brain as being responsible for specific kinds of cognition or cognitive processes? Okay, so again, we are starting at the level of individual neurons, right? So in chapter 1 of your text, Goldstein, the author, defines the mind as a system that creates representations of the world that we can use to act to achieve our goals, right? So the key word in this definition is representations, right? Because what that means is that everything we experience is the result of something that stands in for that experience, right? So everything that we experience, whether it's a particular stimulus or a particular object in our environment, right, or an event in our life that is meaningful in some way, right, our mind, our cognitive architecture allows us to create an internal representation of that stimulus or that event or that experience, which we can use to inform our current behavior, right? So I know that sounds very, very complicated, right? But it's actually a fairly simple process that we use in pretty much every aspect of our daily life, right? So one of, again, the primary goals or the primary functions of cognition is to create internal representations of events, objects, or experiences, right? To relate this to individual neurons, right? What a cognitive neuroscientist would want to find out is how do neurons represent these various characteristics in the environment, right? So everything that a person experiences is going to be converted to an internal representation, right? So how are those internal representations coded by individual neurons, right? So how do individual neurons represent various objects in our environment, right? So that particular question is called the sensory coding problem, right? So again, it's how do individual neurons code for or represent objects in the environment? Okay. So most of the early attempts to address the sensory coding problem were localized to research in the area of vision, right? And a lot of this research was focused specifically on animals. So one of the most famous studies in this domain was conducted in the 1960s by Hubel and Weissel. Okay, so what they did in their experiments is they used cats and they projected various kinds of visual stimuli onto the cat's retina, and they measured the activity or the neural firing or the action potentials that were being generated by individual neurons in the cat's visual cortex, right? So the visual cortex is located in the occipital lobe in the very back of the head, and it is responsible for basic object recognition. Right? So what they want, the question ultimately that they wanted to answer is they wanted to look at the neural firing of individual neurons in the visual cortex, and they wanted to find out what type of stimulus causes this individual neuron in the visual cortex to fire. And what they found as they recorded the electrical activity or the action potentials fired in these individual neurons is that each neuron in the visual cortex responded to a specific type of stimulus that was again projected onto the retina. Okay, so these individual neurons that responded to specific stimulus features were known as feature detectors because they only fired in response to specific aspects of the stimulus, right? So let me show you what that looks like. Okay, so here we have a cat that is being restrained in a harness while individual images of various types of stimuli are being projected onto the cat's retina. And you can't see it in this particular image, but the image in your textbook shows you that there's also an electrode in the visual cortex, again in the back of the cat's head, looking at the neural firing of that individual neuron. Okay. And again, what they found is that each neuron in the cat's visual cortex responded to specific features of the stimulus, right? So there were some individual neurons that responded to orientation, right? So whether the bar was pointing to the right or to the left. There were some neurons in the visual cortex that only fired when this particular stimulus was in motion, right? So those neurons are coding for or representing movement. And there were other stimuli, or other neurons, that only fired action potentials when looking at a short bar, whereas others only fired when they were looking at a longer bar, right? So we have individual neurons that are representing orientation, length, and movement in the visual cortex. Okay. So a related area of research that also looks at individual firing of neurons in the visual cortex of cats is research looking into what's called experience-dependent plasticity. Okay, so again, I'll go through the basic experimental procedure and results with you, and then we'll talk about that particular concept of experience-dependent plasticity, right? So when a kitten is born, in a typical scenario, in that kitten's visual cortex, like I said, it's going to have feature detectors that code for all different aspects of a stimulus, right? Movement, length, etc. Okay, so some are going to fire when something is horizontal or vertical, etc. Right? So it can basically represent, using neurons in its visual cortex, all aspects of basic visual stimuli. Okay, what researchers did in this particular series of studies is they raised kittens in an environment where they were only exposed to vertical black and white stripes. Okay, so these kittens were never exposed to anything horizontal or any stimuli that were horizontal. And so what they did is they looked at both the kitten's behavioral response when exposed to a vertical bar and also the response of individual neurons in the visual cortex. And what they found is that the cats only responded, right? And cats in this experiment responded in exactly the same way that an ordinary house cat would respond when it sees something in their immediate visual field, which is they batted at it, right? So they were sort of playing almost. I don't know why I'm miming it for you guys in the event that that might be helpful. But so they would only show any kind of behavioral response when they saw vertical bars, which were again those they had always been exposed to. But when they saw horizontal bars, they didn't respond at all, right? Similarly, neurons in their visual cortex only fired action potentials displaying, recognition, visual recognition, when they were exposed to verticals, not when they were exposed to horizontals, right? So what does this mean? Okay. Oh, and I should say that they conducted similar experiments where they were only exposed to horizontal bars, and they showed the same pattern, except it was the inverse. So they only batted in response to horizontal bars, or neurons only fired in response to horizontal bars. So here is a picture that just kind of shows you what the experimental apparatus was in this case. So the cat was basically placed into this structure that showed them only black and white vertical bars, and they weren't able to see anything else in the environment because they were wearing that sort of cone on their head to block out all other visual stimuli, right? So what this diagram shows is the orientations or the visual stimuli that showed maximal firing. So it looked at 75 individual neurons in the visual cortex in one of the graphs and 53 in the other graphs, right? And again, it shows that vertically reared cats only showed maximal neural firing when they were exposed to vertical bars, and horizontally reared cats only showed maximal firing in their visual cortex when they saw horizontal bars, right? So going back to this notion of experience-dependent plasticity, right? What this demonstrates is that we have highly specialized neurons in the visual cortex that code for very specific features of stimuli, or represent at a neural level very specific features of stimuli. But it also shows that the visual cortex, neurons in the visual cortex, are highly specialized and highly malleable depending upon the nature of, in this case, the cat's experience. Okay, so the brain can actually change its very structure in response to what it is exposed to, right? And whenever we talk about the brain being highly malleable and responsive to changes in experience, that's something that we refer to as plasticity, right? So this demonstrates a lot of specialization in the visual cortex at the level of individual neurons, and it also illustrates something called plasticity. Okay. So I'm just going to show you a couple more examples of evidence that there is specialization of function at the level of individual neurons. Okay? So in another experiment with a similar sort of protocol, researchers presented various types of stimuli to monkeys and recorded activity of single neurons in the temporal lobe. So again, they're essentially asking the question, what makes this individual neuron fire action potentials? What stimulus do I have to present in order to excite this neuron or prompt it to fire an action potential? Okay, so this was actually kind of a tedious experiment for these particular researchers. In total, I believe it took 3 or 4 days, your book said, to complete this experiment. And basically what they did all day, every day for that period of time is they just presented, right, a number of different kinds of visual stimuli, right? So just like with the cats, they projected these images onto the monkey's retina. So they presented lines, squares, circles, so very simple stimuli at the start that were either light or dark. Okay, and as they were sort of mapping out the temporal lobe of these monkeys and seeing what these individual neurons responded to, they found a neuron that didn't show any sort of neural response at all. So it wasn't firing action potentials in response to any of these stimuli. And it just so happened quite fortuitously that one of the researchers kind of moved his hand such that his hand was projected onto the monkey's retina. And what they found is that there was a sudden burst of firing. So all of a sudden, after the hand was perceived by the monkey or recognized by the monkey, the neuron started to fire a lot of action potentials. Okay. So what this diagram shows is it shows the firing rate of this individual neuron, right? So here are all of the different shapes that the researchers presented to this individual neuron. And what the number below the shape is indicating is the degree of firing of this individual neuron. And it's kind of confusing, but 1 actually means there was no firing. So there were no action potentials being generated from this individual neuron. And 2 and 3 indicates moderate or actually very little firing, whereas 5 and 6 is consistent with maximal firing, so firing lots of action potentials, right? So what they found is there was really almost no response for simple stimuli, but as the stimuli got more complex and more hand-like, there was maximal firing, right? So not only are there individual neurons that visually represent simple shapes or simple stimuli on the basis of individual features, but there are also individual neurons that seem to code for even more complex stimuli. Right? And more experiments with monkeys looking at the temporal cortex seem to support this, right? So later researchers documented that there are many, many examples of neurons that seem to respond or seem to fire action potentials only when they're exposed to faces, but they don't respond to other types of complex stimuli. Right? So whether it is a primate face or a human face, that prompts the firing of action potentials, but equally complex visual stimuli like pictures of nature or pictures of buildings don't cause any firing. All right, so I've shown you guys a couple of different examples of experiments using animals that have shown that individual neurons code for or represent various kinds of stimuli in the brain, whether those stimuli are fairly simple or complex. So what I'm going to do now is share one more experiment with you that actually used human participants. So in this particular study, the population that they were working with was individuals who were undergoing brain surgery for the treatment of epilepsy. So for those of you who don't know, epilepsy is a neurological disorder that is caused by basically too many or too much excitatory activity in the brain. So as a result of damage to individual neurons or even large assemblies of neurons, those neurons come to basically fire too many action potentials, and that kind of causes an electrical storm in the brain, which leads to seizures. So one of the first lines of defense for treating epilepsy is actually to do brain surgery where you identify and remove the damaged neurons that are causing this surge of excitatory activity in the brain. So one of the ways that you do that is you basically map out a person's brain by stimulating various neurons in a particular region of the brain and looking at their rate of responding, basically as a means of identifying or differentiating healthy neural tissue from unhealthy neural tissue. So what they did in this particular study is they looked at activity in the temporal lobe while they were undergoing this basic procedure. And very similar to what they did with cats and monkeys, they presented various images and recorded activity of individual neurons in the temporal lobe. And what they found is that, as I said, like the cats and the monkeys before them, these individuals showed neurons or neural firing of specific neurons that only responded to very, very specific stimuli. So in the most extreme case, they actually identified a neuron that only fired action potentials in response to pictures of the actor Steve Carell. And this particular neuron didn't respond to any other human faces. So I'm going to show you in this graphic, the different stimuli that they attempted to, that they presented looking at the individual rates of firing in this neuron. They presented, of course, all of these images of Steve Carell, and that resulted, as you can see in the graph, in maximal firing by this individual neuron. So that, of course, they wondered, what is it about this image that seems to be prompting this rate of firing? And so they reasoned, well, maybe there's a particular feature that this neuron is coding for, right? So maybe this neuron is coding for famous people or people that have been featured prominently in the media, right? So in response to that, they presented pictures of very, very famous people, you know, Bill Clinton, presidents, other actors, and so on. But what they found is even when they were presented, or in this particular experiment, they presented images of other famous people, there were minimal rates of firing. And so they did this procedure for a half an hour and presented all these different images, trying to figure out what this individual neuron was coding for. And the only thing that resulted in maximal firing were images of Steve Carell. So it's possible that we all have a Steve Carell neuron in our temporal lobes, but it's also possible that if they had had more time, right? So they only did this procedure for about 30 minutes, which isn't a lot of time. As you saw with the preceding experiment with the monkeys, right, it took them 3 to 4 days to discover what these individual neurons might be coding for. So the researchers caution people in reaching the conclusion that we do have a Steve Carell neuron, saying that maybe if they had had more time to present more pictures, maybe they would have discovered something more general that neuron was coding for. But it's still fairly interesting, right? Okay, so this concludes our discussion of specialization of function at the level of individual neurons. Okay, so I will see you guys in my next video talking about the specialization of individual neurons, or individual brain structures, excuse me. All right, so I'll see you guys soon. Bye.